So without any further ado, let me introduce Katerina Kechris. She's a professor in biostatistics at the Colorado School of Public Health at the University of Colorado Anschutz Medical Campus. And she's been working, she has a long list of accolades. She got her PhD from the University of California, Berkeley, and did a postdoc at the University of California, San Francisco. She's an ASA fellow. She has been working in the field of metabolomics for many years, developing methods on the statistical analysis of this really complex data set from a statistical perspective, and has been a leader in that field, doing it, looking at a lot of lung diseases and addiction-related phenotypes using this type of technology. And Katerina has been a longtime collaborator with me. I've really enjoyed working with her for many, many years. She's very intelligent and thoughtful and an amazing statistician and collaborator. So Katerina, feel free to take it away.
Thank you, Laura, for the nice introduction, and to the P30 team for setting up this webinar series. It's been great to see all the variety of topics that have been covered. So let me share my screen. And do that. There. Is that working? Okay, great. So as Laura said, the topic today will be on metabolomics. I've been working in this area for about 10 years or so, more on the statistical and data analysis side. But today I'll give an overview of the platforms and then get into the different data analysis pipelines. As Laura mentioned, if you have any clarifying questions, feel free to put those in the chat, but more discussion type questions, maybe at the end when we have more time, we can follow up there. So remove this here. Okay, so I have this an outline of my talk. I'll just talk a little about, defining some terms, what metabolomics is, then I'll get into the technology types, in particular mass spectrometry, which is one of the more common ones used, and the type that I've had the most experience with. And then I'll get into the analysis methods. So this is a pipeline of the different steps. There's many different steps, and I'll give an overview. I only have an hour, so I can't get into a lot of detail. I think there's enough material for probably multiple sessions and a full class, but at least you get an introduction, and I'm happy to follow up with any of you if you have questions. I tried to provide also references, some reviews, if you wanted to follow up with those as well. So just an introduction on some terms. Metabolomics refers to the study of small molecules in biological or ecological samples. And these can be identified using a variety of different analytical strategies. And they can be both targeted, where you have predefined, so usually small list of metabolites of interest, or untargeted, which is more focused on hypothesis generation and performing comprehensive profiling in the samples of interest. So the goals are to identify what small molecules are in the samples, in addition to then quantifying. So there's the goals of identifying and quantitation that I'll get into later when I get into the analysis pipeline. So just a few words about metabolism and metabolites. Maybe many of you are familiar with this, but metabolism is an important process that there could be a series of different types of chemical reactions. I have some examples here, which could be both the synthesis and the breakdown of metabolites and compounds, and a variety of enzymes that perform these functions. But in organisms, usually there's this, they strive to reach a balance, homeostasis, and metabolites help that balance of sort of the breakdown and the synthesis. Okay? So the metabolite profiles reflect some of these balances or imbalances when you have disease states or other perturbations. So metabolism is affected by a variety of factors. Here I have some of the main factors. Genetics plays an important role. Genetic background can influence metabolic rates, and in the different rates, within variants in a population. Our microbiome, our own sort of individual microbiota can have an important role on the metabolic profile in an individual. And then there's sort of exogenous factors, different exposures, whether that's chemical, environmental, diet, drugs, lifestyle, exercises. All of those can have an impact on your metabolism and your metabolite profile that then manifest in the phenotypes that we observe. So here I have just a summary of some of the main classes of metabolites. You'll see this, I'm using these interchangeably. Small molecules is the more general term, and compounds, and metabolites usually refers to when it's actually identified. And I'll talk a little about that later, what the identification process is. So just so you know, I'll be using these all interchangeably. But here I have some of the main classes. I'm sure you're familiar with many of these. Amino acids are the building blocks of proteins, fatty acids are the building blocks of lipids, sugars, building blocks of carbohydrates, but also lipids and DNA and nucleobases, along with sugars, are building blocks of DNA. So this concept of we've got the building blocks, but then we have the sort of macromolecules that we're looking at it as well.
Katerina, I don't know if you can keep the chat open at the same time, but going back to that previous slide, can you also comment on polar versus non-polar metabolites? How does that shake out as a category?
Yeah, so that's kind of another axis, and different techniques are more appropriate for polarity. And I'll show some slides a little later that you can see some of these axes, but yeah, so I would say that's like another axis on classifying molecules. But good point. Thank you for whoever brought that up. So here I have an example of one metabolic pathway. I work a lot in lung disease, and particularly lung disease due to chronic smoking. So here I have the nicotine metabolism pathway. So you can see all these derivatives of how from the breakdown of nicotine. We often actually measure cotinine as a measure of smoking exposure because it's more stable. It has a longer half-life. So those are the kinds of factors that play into what might be measured and what you'll observe. And so now I wanted to get a little into the history. So the metabolites, have been measured for decades now. Some of the earlier work was the late 60s and 70s, and that required a lot bigger sort of machinery. So it wasn't really practical at a large scale level for individual labs to do. It wasn't, you see here, not till 1998, the term metabolomics was coined, and this is the same time that we saw a lot of the other omics coming out as well. There have been leaders in the group, like the Wishart group in Canada, that was at the forefront of developing these methods and developing efforts to profile at large scale and create resources like the Human Metabolome Project, looking at thousands of metabolites in human, but also looking at drug and food and sort of exogenous types of chemicals and so on. So this has evolved. Since then, I did a quick PubMed search recently just to see the number of omics publications, just to compare with the other more common ones like genomics. Clearly, that's been around a little longer, and you can see I didn't go far back enough, but you can see thousands of publications. Where proteomics and metabolomics came on the scene a little later, but have grown very rapidly in the last 15 years. Metabolomics is here maybe newer, relatively newer. So there's a lot of interesting sort of evolution of the technologies and also the data analysis that have become very rapid. And you can see this sharp increase in more recent years. So a lot of growth in this area. And I don't have time, of course, to survey all of the a lot of the both foundational work, but also all the new direction. But I just have here examples of the translational research, both looking at metabolic profiles here in human, just in normal health, which is of interest, but then also looking at disease, that metabolites can be used as biomarkers for drug addiction, for example. And there have been applications in this many different disease areas, or just looking at just basic physiology, or looking at environmental exposures. So just the field is growing very rapidly, and a lot of new applications, and a lot of also integration with other types of omics. So here I have sort of summary. I've mentioned some of these already, but some of the key applications I work on a medical campus, so I'm not exposed to this very much, but there's actually a lot of work in the agricultural sciences, a lot of work looking at metabolites and profiles in plants. So a lot of activity there. Here in the medical field, that we see more work related to disease mechanisms or biomarker discovery, nutrition, physiology. I mentioned some of these, the microbiome, toxicology, and then other environmental exposures. I mentioned also sensing. The lab that I work with mentioned they look also at how the metabolome profile changes just based on simple environmental changes like temperature. So the metabolite profile reflects many of these changes. So and sometimes it's of interest to study it because it's considered to be closer to the phenotype. Okay, so it'd be reflecting the changes in the metabolic pathways. So these applications, what kinds of questions can be answered? The most basic is what small molecules are even present. Okay? So that's the identification part. But then when we look at what you can quantitate, then you can see, are there associations? Are the metabolite levels changing with the disease spectrum, with the exposure, diet, and chemical exposures, and so on. But there's also of interest in these sort of precision medicine approaches to think about the predictive power of metabolites for diagnosis. So I do a lot of work with biomarker detection in lung disease using metabolite profiles. They can also be used to study mechanisms. What is this? What are the active components of the pathways, and what pathways are relevant in that context? I mentioned genetics. We've also done a lot of work with looking at variants that are associated with with metabolite levels. And there's some very strong associations where a single SNP can have a very large influence on the metabolite levels. So we've been cataloging those and then trying to link them with disease and the same metabolites and how they affect disease severity. I mentioned the microbiome, a lot of interest in that more recently. So often groups profile both the microbiome and the metabolome in parallel and try to understand the role of the microbiome on metabolism. And with the right study design, the right context, you can also look at metabolic flux. For example, what is the rate of turnover of molecules through a metabolic pathway? Okay. So this would be specialized type of analysis. It wouldn't be the standard sort of untargeted approach, but if, again, in the right study design and context and sample, you can also ask these kinds of questions. So to make things a little more concrete, I wanted to show you one study from our group with pulmonologists who study chronic smoking. And we had an animal model. We were looking at mice and it tried to examine their sort of gene and metabolite response over time to cigarette smoking exposure. And we were looking both at the lungs and the blood from the mice. So I just wanted to spend just a couple of slides. I can't get into a lot of detail, but just to show an example of a study. So these were mice that were put into a cigarette smoking chamber. And we follow them over time. So there's sort of acute stage. You can look at differences, changes in one day or the first week, then looking at more immediate chronic sort of exposure, and then finally long-term sort of chronic exposure. And we had 5 mice per time period, and we compared those with their controls, and also looked at a cessation model where these were mice that were exposed for 6 months, but then the last 3 months they were not exposed anymore to cigarette smoke, but in the air control. Then we could look at reversal of the effects. So this was both a gene expression and a metabolite study. Here's some of the key pathways. And what I have here is in color sort of our pathways that change up, where a lot of the genes are up or down regulated in the pathway in color. And then the checks are related, or when the metabolite profiles also showed changes for that pathway. And what was interesting is that we saw some immediate sort of response, but sometimes those were altered. And actually, when the smoking exposure became more chronic, but then in the cessation, you saw the reversal. The 3 to 6 month time points that had the sort of weaker signal, but definitely the earlier in the chronic time points showed the most changes. And you can see is that a lot of these pathways were supported both by the gene expression and the metabolite profiles. And I wanted to focus on one of these, pyrimidine metabolism, which here I showed. I know this is a very busy slide, but these are examples of these pathway diagrams, and I'll show you later some links to different databases. In squares are the different enzymes in the pathway, different other proteins that are involved, and color-coded are whether we see down regulation. They're mainly green in this case due to cigarette smoking exposure. And then in circles are the metabolites that are involved. So this was just to show an example where you can start of an application and how you can also link different omics data sets, because there's very much interest now. You can get different profiles and different snapshots of your system by using the different profiles. So that is just a quick summary of sort of introducing some of the topics. Before I go to the technologies, and now unfortunately I can't see the chat, so I don't know, Rob, if there was sort of a clarifying question, I'm happy to answer it, or I can move on.
One of the questions that got brought up, Katerina, is does the Wishart data for human include brain or brain regions?
I haven't looked at it recently. That's a good question. I believe they do have different sample types. That original resource has grown now. That was over 13 years old. So I don't know specifically. You may want to just check, look at the human metabolome database.
Just maybe a broader question, Katerina. What do you consider the biggest, meanest animal model or human metabolomic study with true genetics in it?
The biggest.
Any.
Well, I don't know. We've done one. It just came out recently in the last couple of months. We were looking at chronic obstructive pulmonary disease and had a cohort of, these are older individuals with chronic smoking history, chronic or former smokers. And we found very strong, at least.
But that would have been blood, right?
In blood, yes. Okay, so that's what you're asking. Yeah, this was in blood.
This is NIDA, so we're primarily interested in what's happening in the brain.
Yes.
And I challenge anybody here to find a single metabolomic study of brain of any species, Drosophila. I'll even give you credit for Drosophila. That includes more than, say, 100 samples. And that I would love to be proven wrong, but I don't think it exists.
Okay, no, I'm not aware of that, but I haven't looked recently. So if anybody does find it in the next hour, please share that with the group.
It's a huge embarrassment, if you ask me.
Yes, I see.
We've had these methods for close to 20 years, and nobody's done a genetic analysis. But
yeah, most of the ones are blood. I agree. They're not just because of the convenience and the ability to get the large sample sizes. But yeah, thank you. Okay, if there aren't other questions, I can move on. So the next section is a little about the technologies. This is a quick survey because I do want to spend more time on the analysis. Before we get to the technology though, we were just talking about blood. What are the most common sample types? Of course, blood is most common, plasma or serum. Serum is plasma minus the, some various factors. Often urine is used, but anything is really possible. Saliva, stool, tissues. I mentioned, the lung samples in the mouse study we did. Rob was challenging you to find a big brain study. Like I said, anything is possible. There's some considerations because this is related to metabolism. You have to think a little about, again, if you're looking at human fasting, that can affect the metabolite profiles, the time of day. Hemolysis, which is the rupture of red blood cells. If you're looking at blood, we've actually had that kind of contamination, and did it affect our results? Freeze thaw cycles, and some of these are general; they're not specific to metabolomics, but it's good to know, what this preparation and the sample collection, and because all of these do affect the profiles. So once you have the sample of interest, here's a summary of the main technologies. There's nuclear magnetic resonance, NMR, and then there's the mass spectrometry, MS-based methods, either liquid chromatography or gas chromatography. This figure just shows sort of a summary of the instrument detection level by the number of metabolites, and there's usually a trade-off where I'll go into some of these in more detail next, but there is a trade-off in how many metabolites you get, whether it's targeted or untargeted, and your detection and sensitivity. So NMR, sometimes you may actually see that referred to as Metabonomics, and it's very subtle. There's one letter change, but it's usually specifically referred to for NMR-based metabolomics type of measurements. NMR has some strong advantages. It can handle intact tissue, so it's not destructive. There's no separation steps. It's also considered very highly reproducible and relatively simple sample prep, and it can provide absolute quantitative data. So those are a lot of the advantages. In addition to, it can also be linked with imaging-based methods and looking at metabolic flux. So for certain types of questions application, it's really the go-to. But for sort of larger untargeted hypothesis or generating types of methods, it's not ideal. It's less sensitive in terms of the detection limits, and also it can only measure a much smaller limited number of metabolites. So for those untargeted sort of comprehensive type of profiling, it's not preferred. The mass spectrometry technologies are referred to. I'll list these here quickly, but I'll talk about them a little later. Gas chromatography, liquid chromatography are the most common. LC-based methods are more popular now. But compared to NMR, these methods are destructive in that the sample is used. So if that's an issue, then NMR is probably the better choice for you. Right now, the most common method is this ultra high pressure LC-based method. So in a few slides, I'll talk a little more about these, have some graphics. But before I go to that, I did want to make the distinction between the targeted and untargeted. Many groups use, some are more relevant. If you know what you're looking for a certain class of metabolites, then targeted is a way to go. And we often link we've done both targeted and untargeted. So targeted refers to looking for a small group of known and characterized metabolites, where the untargeted is basically a comprehensive coverage. You try to find whatever's there in the sample, and it's only limited by sort of the machine techniques. So targeted would be sort of more of a hypothesis testing. You've got a small handful. For example, we did for our cigarette type studies, we looked at sphingolipids and had a targeted assay of 69 sphingolipids. So we were specifically testing these sphingolipids. Well, the untargeted is more of a hypothesis generation. We don't know what's there. And it could generate hypotheses about different pathways and different classes. The targeted, we know the identities, we know what the metabolites are. With the untargeted, we have to predict them, and that's actually a challenging problem. Untargeted provides absolute quantitation, while the untargeted provides relative quantitation. So the targeted, just a couple of slides on that. Here I have a picture. This is multiple reaction monitoring approach, where you may have some precursors and a mixture of metabolites, but the ones that are selected of interest. So here's one that's selected of interest that's then proceeded through the process and fragmented, and you have product ions. And to quantitate them, they use internal standards for both the identification and quantitation. So the quantitation, you have these so you have internal standards for the metabolites of interest, and these are labeled. This is heavily labeling, and then they're compared with the samples, which are the endogenous metabolites in the sample considered light. And then, so the heavy labeled standards, those are spiked, sorry, the heavy standards are spiked in at known concentrations. And then that can be used then to measure, when you have the relative intensities of the endogenous metabolites, to calibrate the concentrations. And that's how you get these absolute quantitations, which is not possible on the large scale for the untargeted approach. So that's the advantage of doing these targeted assays. So the untargeted, again, I mentioned this already, it allows for novel discovery, comprehensive coverage, but there's a lot more data. It's more complex to process, and there's this difficulties of identifying unknown small molecules. And with, you think of human samples, you have both endogenous and also the exogenous, drugs and chemicals. The Also, there could be issues with the coverage of the platform and the limit of detection, and that there could be a bias towards detection of high abundance molecules. But often untargeted analyses are followed by target analysis to validate. Okay, so there's not an either or, these could be used in conjunction. And like I said, we've published papers where we've had both. So before I talk in more detail about the mass spectrometry technologies, I did want to say one distinction here on lipids. Sometimes you may see lipidomics used separately than metabolomics, and that's why, and that's because lipids, profiling them requires some sort of separate type of sample prep or analytical method and data processing, because lipids have many different variations of length, carbon chains, and bonds, number of bonds. And so that leads to their own characteristics, mass spectra, the data, and needs to be leveraged in the data processing. So here I have this example here, and maybe this is go back to that question before, is metabolomics here. I mentioned there's sugars and nucleic acids and amino acids, and here are the lipids. Here's some of the main classes of lipids. And there's also this distinction between polar and non-polar as well. So lipidomics is specifically focused on lipids. And it requires, like I said, it's on both the experimental side and the data analysis side, specialized pipelines. Okay, so now I wanted to go into more of the mass spectrometry, and if there aren't questions, I can kind of jump into that. So I mentioned there's this chromatography step first. So I wanted to just have a slide just to get a little more detail on that. So this is some of the sort of sample prep part before the mass spectrometry. And for those of you who work in proteomics, it's a little different with metabolomics in that node. Often for proteomics, you need to do some kind of digestion to obtain peptides of proteins. There's no such step like that here, but there is this chromatography step where you have a sample, and it's important to spread out the analytes over time, because, and there's a next step of ionization, it can be sort of overwhelmed by large quantities of an analyte. So that's what this figure here shows. There's this, insert the sample mixture, and then you have the stationary phase where the analytes separate through this column. The mobile phase carries the analytes through this chromatographic column to the mass spectrometer. And this mobile phase can be gas or liquid, and that's that distinction. I introduced that a little earlier. You can have gas or liquid chromatography. Liquid, from my experience, seems to be used more often. So now we get, the analytes are now ready to go to the mass spectrometer. Here are just some basic principles. There's this first ionization step where the atom and molecules need to acquire some negative or positive charge right here. In targeted assays, only ions that have been pre-selected are the ones that are sent forward, where the untargeted, all ions are read and move forward for detection. There's an acceleration phase where the ions are accelerated, and then later they're deflected through a magnetic field to then reach the detector. And then these beams of ions are detected electrically. So there's different types of detector. And if any of you read a metabolomics paper, you may see these names like time of flight or Obertrap. That's what that's referring to. But the important point is the detectors need to have high resolution so that there's enough decimal points to be able to determine molecular formulas. And that's some sort of, as these technologies evolved, that's what they've improved on. Just a few words on that first ionization step. Here, I'll go back, this step right here. Because again, if you read a metabolomics paper, you're going to see some these acronyms. So I just wanted to define them. So acronyms like ESI or MALDI, PPCI, they refer to the ionization step. And going back to the polarity issue, some of these methods are better for different levels of sort of polarity and molecular mass. The core that I've worked with, they say that this ESI one is the most popular. Again, just in case you ever read a paper, you can hopefully what these terms mean. So now we finally get our data. So this is actually multidimensional. We have the retention time. This is the time for the solute to pass through the chromatography column time from injection to detection. But then there's also the mass to charge ratio. That's what's detected in the mass spectrometer. And then you get these curves, these mass spectra. Okay, so there's 2 dimensions, retention time, mass to charge ratio, and the level is the intensity. And that's what will tell us about the relative abundance of a metabolite. So this raw data is very large. And actually, sorry, before I go to, I wanted to mention one thing. You can collapse in different ways. This chromatogram here is sort of the mass spectra collapsed so that so you can summarize this across retention time. And that can tell you a little about the use later for the quality. But also you can look for fixed retention time, looks across the mass to charge ratio and look at the intensity. So this is what the raw data looks like. It's a little different than some of the other omics. For like RNA-seq, you don't have the 2 dimensions that you do. You have sort of genes or your feature of interest and then count. Here we have the 2 dimensions and then we have the intensity. But these raw data files are very large, often binary, often proprietary too, because they come out with the commercial software. But there are converters to try to get open source formats, so you can use open source software as well. So you'll see both of those used. So now we have the raw data, and I want to get into the analysis method and the various steps. I can pause for a second if there's any questions, or I can jump in.
No questions in the chat at this time. Okay,
great. So here's an overview of the pipeline. So we talked about the samples, we talked about getting the data, the spectral data. So I broke it down into 4 steps here. There's the spectral processing of the raw data. And that creates a feature table or matrix that, so I'll refer to this as a pre-processing, is using look at the spectral data. And then often, once you get the feature matrix, you sometimes still need to perform some additional processing and to prepare it for data analysis, for statistical analysis. So that's the pre-analytic stage. Then, at this point, these are only labeled by their mass to charge ratio, retention time. We don't know what metabolites they are. So that's the identification step. And that can be done in parallel with statistical analysis and any other downstream analyses. And then, of course, pathway analysis is part of the downstream steps, but this would require the identification. So these can be done in parallel, but pathway analysis is usually must be done after identification. So I'll walk through each of these. So pre-processing and pre-analytic. So pre-processing, we want to generate a list of compounds by mass to charge and retention time with an estimated abundance, and this is for each sample. And I'll talk about some of the 4 main steps. There's a lot of variations of this and other depending on the platform, and occasionally other steps as well. But at least you'll get a flavor for what needs to be done. Here's that original figure that I showed. There's some initial data exploration to look at overall patterns and identify problematic samples. This is true of other omics as well. So I mentioned the chromatogram building. That's where you collapse in that mass to charge ratio, that's to retention time. So that's the blue line. And here you can look across samples. And basically you want to see if samples are behaving similarly. I would say here they are, they're shifted a little, but that's okay. But you don't have one that looks very different. Also, you can look at overall intensity if you have the mass spectra across the different signals and peaks. Here's across samples. Again, these are generally kind of in the same ballpark. Again, you wouldn't want to see one sample that's very different, high or low compared to the others. But then there's other steps. If you zoom in on these spectra, you can see that sometimes there's some distortions. Sometimes the assumption is that we were trying to look at the peaks, but if there's some background levels, some background noise, that can mask that signal. So there's some baseline correction methods where you want to adjust and subtract any kind of baseline noise so that you get something that's sort of flat, and then the peaks will hopefully then signify true signal and true intensities. Peak detection. So this is one of the more important steps. So if you look on the top right, you'll see retention time here on the x-axis and mass to charge ratio on the y-axis. And then the color is that intensity. So this is a version of the graph I showed before. So mass to charge axis tends to be a high resolution and doesn't shift too much, so it's a little more stable. So what you can fix that and then look across the retention time and find here this region of interest, the ROI, that has relatively higher intensity. That's a yellow. So now this is this region of interest, and the idea is how many peaks are there, and each of these peaks can denote a possible metabolite or form of it. So there's some sophisticated methods. I don't have time to get into this, but they use wavelets, and they do convolution and peak detection approaches to find now in this case there were 3 peaks. Okay, and those are color coded differently, and those are potential unique metabolites. And what's returned is the retention time. So some may be average or center point, the mass to charge ratio that was here, and then some kind of integrated intensity that reflects the abundance of the metabolite. Okay? And that's done for each peak. So this is for each individual sample. Now what if we have multiple samples, but we're doing this peak detection on each sample. So the next important step is peak alignment. But that could be challenging because the time of evolution can vary across samples. So before, if you look at these different samples, those are the different colored lines, the peaks don't exactly line up. So alignment refers to finding the corresponding signals across samples. So now you can say, oh, this is the same metabolite for all the samples. The problem is that sometimes these shifts are nonlinear. It's not just a matter of just going over left or right a little. It's sort of the shape of the curve changes. So there's different ways to align. You can use the spectra themselves, and there's these sort of sophisticated methods that can stretch or shrink called warping methods that can capture nonlinear transformations to try to then find a good alignment. They can rely on some fast Fourier transform methods, or you can use the peaks themselves. Where you have, I showed that picture before, we have the peaks here, there's some kind of boundaries, and those can be used to align across, each of the samples has their peaks. And you can find the boundaries of the peaks and then line them up across the samples. So these could be done at different stages. There's pros and cons with the different methods. But now we have something, now we can compare, is this metabolite, how is the abundance changing across samples? There's some quality steps that are important. For example, here are the peaks, one peak across samples that are color coded. So here's the mass to charge ratio, here's the across retention time. You can see that in general, the peaks fall on top of each other. Maybe they're different heights, and that's okay. That's reflecting different abundances in different samples. So that would be considered a good peak. With this one here on the right, you can tell it's very jagged. There's multiple modes. There's even this part right here. This would be an example of poor quality. So these would be the kind of peaks that would be filtered out and be considered low quality.
Katerina, can I stop you for a question?
Yeah.
In the chat. Sure. The question is from Milad. It's, do you align the same modes of the wavelet across samples or the combined?
I don't, so let's see, you mean over here? If you combine, well, there's the peak, there's the alignment of the peaks themselves. That's that can be done at a later stage. Or there's the alignment of the spectra. I think, is that the question that's, I think, being referred to here?
Milad, feel free to unmute and clarify if you'd like.
Yeah, exactly. I was curious if you align the spectra in different modes differently or the combined wave.
Yeah, I think there's different methods. I don't know especially what this one does. I think they use these fast Fourier transform methods. I think it depends on the method. So I'm sorry, I can't answer specifically. I think some do use the spectra directly though.
Sounds like a perfect machine learning environment.
Yeah. So, and the point is that you want to capture these nonlinear transformations, and that's why it can be very complex. These are usually taken care of by the cores that we work with. They have more expertise because they do this on a regular basis. So this is often the cores do these steps in the alignment. And like I said, there's, I think CentWave was the one algorithm here that I mentioned before, and I'm happy to follow up with some resources to answer your question more specifically. Okay, other questions. I wanted to talk also about, now we're thinking about across samples. So instrument, their response and their sort of measurements can change with time. So there's going to be drift that occurs. And this, especially you see this with large batches or samples that were processed in the same study, but over a longer period of time. So here I have an example of, these are each samples, they're color coded by batch. And there's a principal component analysis of the entire sort of profile. And you can see clearly here that the colors are grouping and there's strong batch effects. And sometimes in our experience, for example, this is separated, for example, by the weekend or, even sort of time period, you can see which batches were performed when during the duration of a study. So it's important to normalize because, and this is common true for other omics studies. Sometimes there's simple normalization methods like just taking the sum or median signal intensity for each sample and normalizing so that they're all approximately the same. That makes some assumptions that they're all, that the same, all metabolites experience the same pattern of drift. That may not always be true. Cases where there's internal standards, those can be used, but that's usually not so practical with these untargeted approaches, since you don't have internal standards for all the metabolites. I think what's used more commonly are quality control samples. So if there's multiple batches, these are sort of the technical samples that are repeated. And they're, so you can see here, this quality control sample in the sort of different time periods, so in the injection order going across, and you can see how the intensity changes. So it's pretty dramatic. And then when you look at the raw data, so that raw data would be the other samples, the samples of interest, those are in blue. So they kind of follow this trend as well. So the idea is you can use the QC samples to predict that trend and then adjust for that trend. So here it's, this is a paper that did different approaches. They just did a linear or more of a smooth base, so it uses smoother. But if you adjust even with the linear, then these samples then start to become more similar. And they show this, this approach performed the best in their comparisons. So these QC samples are important for these bigger studies to adjust for these drift. Okay, so now we've performed the peak detection, the alignment, we have abundance measurements, we've adjusted for any potential drift. And now we have a table of potential metabolites, but they're still sometimes not quite ready to perform statistical analysis. You still need to do some pre-analytic steps. So I'll go through some of those. So one step may be if you have replicates, that's not required, but sometimes there may be duplicates or triplicates, so you need to summarize those. So these are technical replicates for the same biological sample. So this is something we've come across with some of our data sets. So we've actually developed, we have an R package that can process, how do we summarize replicates? One thing is to look at the coefficient of variation across the replicates, and depending if it's low or high, you may want to use different types of summaries. So if it's low, that means the replicates are pretty consistent. You just use the mean, or if it's high, you may either want to remove those observations, or take the median, which is more robust. So here I have an example of actually data that we've worked with. This is not long transformed yet, but here's one peak in one compound where we have replicates that have been sorted by low, medium, high, and you can see how one of them is much more variable. So the coefficient of variation would capture that. And in that case, you may want to take the median. So the replicates are not always used, but if they are used, it's important to either summarize them or model them later as repeated measurements. Then another sort of feature of metabolomics data is missing data.
Can I just, so on the comment on replicates, were those technical replicates or biological replicates?
Sorry. Technical replicates for a biological sample. So
okay, got it.
So usually duplicates or triplicates. So now we get to missing data. So there's actually you see a lot of missing data in metabolomics data sets. There can be for different reasons. First of all, the compound may just not be present in the sample. That's more of a biological reason. For example, in our smoking studies, some individuals were former smokers. They weren't smoking at the time, so we wouldn't expect nicotine sort of derived metabolites to be there. So that's more of a biological reason. Other reasons would be more technical. For example, the compound's present but below the detectable limit of the machine, or within some specified noise level. So that's more of a technical reason. Or it may just be failed to be identified. So that would be maybe an error in the sort of the assay and the mechanical error in the lab prep, or through the chromatography or the coelution, more of a, again, experimental reason, or in the bioinformatics later. So I remember I showed you how to get peaks and how we do the peak detection and peak alignment. Well, those rely on informatics methods and some assumptions and some algorithms, and that may have some errors. So that would be errors in the pre-processing. So these modes of missing in statistical language can be described as missing completely at random. That's when it has nothing to do with the level of the metabolite. So that would, as opposed to missing not at random. So a compound being present but below the detectable limit would maybe be missing not at random because it's missing because it's low level. Okay. Versus something that's at random, well, maybe one of these like mechanical reasons or the technical sort of experimental reasons may be missing completely at random. So this is actually this paper that had some nice sort of visuals of what you would see. If it's missing completely at random, if your samples and metabolites, you would just expect X's are missing, just randomly distributed. If they're missing because of below the limit of detection, if they're sort of here ordered, this metabolite's ordered, you would expect all the smaller ones to be missing, or smaller values. But in most data set, it's some combination. You've got missing at random and missing not at random. And it kind of, and here, this is probably the most realistic, where it's probably some combination. And it depends on the metabolite. Some metabolites that have high abundance probably won't have this lower limit of detection problem, but they would have missing at random. So it gets complicated, and you don't know what missingness you have.
Katerina, there was one question there about that slide from Shauna, actually, that was using the term censoring. So is that missing or censored or left censored or?
Yes, so we assume left censored, that it's missing because of the lower, because it's small values, yeah. So people have tried to model this, and that's actually our group. Do we try to model, in this case, as censored data versus the missing completely at random, where that would not be the right assumption. So how do we handle this? Because a lot of the downstream steps, when you try to do statistical analysis or machine learning, the missingness causes problems. So some people often do is, first of all, you may just remove compounds that have high levels of missingness, that you want to see them with at least 80% of the subjects. There's some data-driven approaches. I think this would depend on your study. For example, for our smoking study, well, half of our subjects were current smokers, well, half were not. So maybe that 80% would not be a good cutoff. So the advantage of doing this, it removes some noise detected as compounds, some uncommon food or drug metabolites. Maybe some person happened to have, take some, medication that nobody else has. So that would remove those kinds of metabolites. It also simplifies the downstream analysis, that you're not testing as many metabolites. Your dimensionality has been reduced, but it also has some, can have some negative impact on the downstream analyses. If you filter too much, you may be missing important compounds. Like I mentioned, the smoking related one. If you're too lenient, then increase, like I said, you put in these potential false positives, you put in some of these rare drug or food metabolites that maybe that are you wouldn't want to test. So you increase your multiple testing burden. So this is, you'll see some guidelines in the literature and sort of some standard practices, but you may want to be more specific to your study.
I just want to add, Katerina, that this makes me really nervous in a genetic context, just because the best case scenario is 50% of your population is null and 50% has killer expression, and you've got a beautiful bimodal, extreme bimodal distribution. And those are cases where if you have a threshold and you say, I'm going to throw anything away with, X% missing, you can sometimes throw the baby away with the bathwater. So you just have to be aware of that. So I prefer to put the data into Gene Network in two forms. One, the whole kit and caboodle, and then, however you want to clean it up for a particular application or paper.
Yeah, I agree. I think you would not want to have, like, the data that's in databases should not be filtered. I agree, so that individuals can do their own, filtering. And I want to say there's this newer paper that was published recently that I call it data-driven here, that they tried to look at the quality, use some of the quality metrics to make decisions about filtering cutoffs. So I thought that was maybe more of a data, or, specific to a data set, rather than having these, rules that may not be good for every single data set. So, but I agree. I don't think you should. Yeah, you would want not putting filtered data into the database. So that filtering doesn't necessarily remove all missing values. There could be compounds that still survive that have missing values, and some downstream analyses may need complete data. A lot of machine learning methods. So it is common to impute missing values. There's some strategies. One is, no imputation, but a lot of methods and statistical assumptions may not be valid. One thing we do is sometimes we make the data binary, like present or absent, if there's a lot of missing values, so that you, for example, like the smoking one, the nicotine derivatives, we just want to know if they're there, yes or no. That's a way to you don't have to impute. The more common strategy for imputing is using the minimum value. All missing values are replaced with, for example, half the minimum observed. This I find, unfortunately, is used very frequently, but I find it problematic. First of all, you're imputing sort of the same value multiple times, and you could artificially reduce your standard deviation and your estimates of variance and so on. So I would proceed with caution with that. There's some better methods out there now that are more holistic. They use more of the entire data set. And based on random forest, k-nearest neighbors, Bayesian principal component analysis. Here's an example where, here you have data from metabolite. It looks somewhat Gaussian, and you've got a big spike with missing values. The minimum value would kind of create this kind of longer left tail, where some of these methods that integrate more of the distribution of the data would give a smoother type of distribution. So usually, random forest and k-nearest neighbors are commonly used. So I would recommend those. So finally, sometimes there needs to be some additional normalization or batch effect correction. Although these QC samples can adjust for drift, there may be additional variation due to other sort of unmeasured factors. So if there's spike in internal controls, maybe those could be used, but there's also more data-driven strategies. There's a batch effect correction. Some of this draws from the microarray gene expression fields, and can still be used to do any additional adjustment. Here I have an example of this batch. This is one of our data sets. Well, I showed something similar like this before, but batches are the symbols and the colors, the disease stage, and you can definitely see the differences are due. I think this was, separated by a week where this study, definitely a time effect. But so there's, strategies for normalization. Maybe I'll go through these quickly because we're getting close to the hour. Some median normalizations tend to be simplistic, just shifting by median or some kind of quantile, like the 75th quantile. But then there's more sophisticated methods. that have, again, drawing from the genomics world, transcriptomics world, but kind of being more specific to metabolomics. Here's some of these latter two ones are more variations that are specific to metabolomics. In the interest of time, I'll just kind of show you, after normalization with one of these methods, you can see now you don't see this batch effects that you did before. So now I wanted to get to the downstream steps and identification. Identification is one of the big sort of challenges in metabolomics research. The idea is we actually don't know. Right now, we have everything was labeled as the mass to charge ratio retention time. We don't know what the compound is. So that's what identification refers is what's the most likely compound and maybe metabolite that's appearing. And this can be done any time before prior pathway analysis. It's usually using the mass to determine that. But the problem is there could be multiple matches and scoring algorithms to define that. So it's a major challenge in untargeted metabolomics. There's various databases that can facilitate metabolite identification. And they have this variety of information. Maybe I'll just show you here some of the main ones, like HMDB. BB is the main one, Medlin, very commonly used. Then there's other ones that are specific to lipids. I have some here, some numbers, but some of these just depend on the curation and of the databases. Here I'll show you one example. We've studied single lipids a lot in our work. So ceramide is a kind of single lipid. I just wanted to show you an entry here. It has the chemical structure. It links to the full names, the mass, links to other databases. So this is the kind of information you would get from these databases. So going back to the identification, what I've actually talked about is the lower level of identification and the least validated. We just have a mass and a retention time associated with a feature, but that can be used to get a molecular formula like this, which is not necessarily specific because this molecular formula can map to multiple metabolites. So that would be considered the next level, be some kind of preliminary identification based on this molecular formula. But to get to the higher tiers, which is the most information, you have to either, you have to look at the spectra themselves. So that's what you can match to these MSMS libraries of the spectra. And for example, HMDB has spectra that can be matched in. So that gives you now imputative ID. And to get to the highest level, you usually require some kind of other evidence, some references or some targeted follow-up. So that's not always possible, but ultimately that's where we want to get. That's the most confidence in the identification. So we get things usually in these kind of intermediate stage. Sometimes all we get is this. It could be some feature that we can't identify what it is using any of the resources in the data in the matches. Often we do get in this range and this at least a preliminary identification or putative. But for example, a data set that I've been working now, about a quarter are not considered sort of identified. They're still kind of at this level, which is improvement. I know maybe years ago, I would get data sets, maybe that half were not identified. So I think there's been a lot of progress, but this is the challenging step. So actually I had a couple of slides showing this. I know I've gone over the hour. I don't know if maybe I could skip. Laura, do you suggest maybe skipping? And I just had some examples here and just getting to the downstream analysis. The downstream analysis at this point is actually like things you would see with other omics. You do various t-tests, regression, ANOVA, depending on your study design. Think about multiple testing. You would use a variety of machine learning methods. I think maybe some of these hopefully have been introduced in some of the previous webinars. These are not necessarily specific to metabolomics. A lot of the other methods work. I wanted to show you some resources. I just have a few more slides. Metaboloanalysis is a really great resource. That's actually originally from the Wishart group. It's really well maintained, and it has a ton of different sort of tools and different stages. And it has R actually in the background, very user-friendly. Here are some of the pathway databases. So I wanted to talk about pathway-based analysis. CAG, that was one of the pathways that I showed in the beginning of my talk, but there's other databases. And I guess the number of pathways kind of reflect the amount of curation. If it's small, more pathways tend to be less curated. And then enrichment. So if I would say if I have my metabolites and I want to see if they're different between certain disease groups, and I find a list of 100 metabolites, there's some enrichment analysis. Are those metabolites in certain classes? Are they lipids or amino acids or other chemical sort of properties? There's methods like this, ChemRich, which is out of UC Davis, that can be used for enrichment. And then there's the pathway-based analysis. This is out of Metaboanalyst, where here's an example of a pathway. And we want to find, did we find a lot of metabolites relative to background that are in this pathway? So did we find a lot of metabolites related to this glycine, threonine, threonine metabolism? What's interesting about this approach is it doesn't just give you a p-value, it also sees are the metabolites that you found changing in your experiment in important parts of the network, or of the pathway network. That's what this pathway impact is. So for example, they would be, you would get more weight if it's kind of a hub in the pathway, so that you get two sources of information, like the enrichment analysis, the y-axis, but also the impact of the metabolites. So you would want maybe pathways that are sort of, so each dot here is a pathway. So yeah, this is all on Metaboanalyst, interesting tools. Just the last two slides, I wanted to mention there are databases. So I think Rob had a challenge to find some data sets. Metabolomics Workbench is one that's funded by NIH. It's part of the sort of Metabolomics Consortium, where they have over here almost 1,400 publicly available studies. So this, for example, if you publish a paper, you would want to upload it here and have you have an ID. So it's kind of like the NCBI GEO or SRA databases for gene expression. There is also the European equivalent called Metabolites. So those are sort of the two main databases for data. And I just want to put a plug. We've been cataloging metabolomics software because a lot of them has been generated in the last few years, last 5-10 years. So Metabolomics Workbench has, if you're interested in metabolomics software, there's this link that can tell you different software for different purposes, and also to generate some potential workflows, because as you saw, there's lots of steps. So this is my last slide. Sorry, I went a little over. I just liked it because it has a pretty picture. This is from Gary Schustak's group. He's a leader in this field. And I liked how he called metabolomics the super glue of omics, which I thought was a nice summary. So